Welcome to the General Pathology tutorial of 2020. Get your white coat and gloves on and scatter those cells yourself. This is the first-person tutorial in Pavia, and it's a bit like Minecraft, but better. We really try to present you with two different experiments that you would have done yourself if it was not for the coronavirus crisis we're in. So with this special format, we hope you can still enjoy doing the experiments yourself. And even though it's not in 3D, we did try to provide you with a quality full HD experience. And this was produced by Dr. Luisa Iamele and myself, Hugo de Jonge. Enjoy! The experiment that you are going to do today is the MDCK cell scatter assay. And this assay tests the effect of the hepatocyte growth factor or scatter factor on highly sensitive epithelial-like cells called Madden-Darby canine kidney cells, which were isolated in 1958 by two researchers, Madden and Darby, from a Cocker Spaniel kidney. And those cells have been crucial for the discovery of the growth factor, the protein HDF-SF. In fact, the name SF comes from its scattering effect on these cells by Professor Gherardi in the mid-1980s. The effect is mediated by the tyrosine kinase cell surface receptor MET, and the observable effect of stimulation of MET in this assay is the scattering of the cells, the MDCK cells, that without stimulus grow in tight colonies. I will not go into too many details and make you lose interest in doing this experiment or even tempt you in fast forwarding through this section. But I do want to mention what happens when HDF-SF binds the MET receptor. When it happens that HDF-SF binds a receptor, it brings together two of these receptors in close proximity. And on the other side of the membrane, so within the cell, tyrosine kinase domains are brought in close proximity, which then start to cross-phosphorylate specific tyrosine residues. And these tyrosine residues, when phosphorylated, start to form specific protein binding sites. And those binding sites are attracting adapter proteins such as GRB2, CRK and GAP1, which then lead to additional proteins being attracted and being activated by the receptor. So we get a whole cascade of phosphorylated proteins, and we have activations of multiple pathways that eventually lead to the up and down regulation of specific genes. And when that happens, biological processes such as actin cytoskeleton modification start to occur, leading to a change in cell polarity and motility, but also proliferation of cells and cell cycle progression, as well as the changes in the way the cells attach to each other, so changes in cell junctions, which leads to migration and invasion, and last but not least, an anti-apoptotic and pro-survival phenomena, which is stimulated by MET and HDF-SF, leading to survival of cells in rather stressful conditions. And why is this important? This is extremely important because we generally believe that 90% of the cancer deaths are actually caused by secondary tumors that occur after metastasis takes place. The spreading of tumor cells through our body using the bloodstream or the lymphatic system, and these cells establishing themselves in different tissues and different organs where they cause big problems. While a single tumor, a primary tumor, can often either be surgically removed or treated for instance with radiation, the moment a metastasis occurs and cells spread to different locations and find different environments to grow new tumors, we're really left with very few options to fight these new tumors. Lung cancer of epithelial cells, therefore called the carcinoma, is an example of a cancer that has often already metastasized by the time the initial diagnosis is confirmed. But also pancreatic cancer and liver cancer are examples of very aggressively metastasizing cancer types. And HDFSF and its receptor MET play an important role in many of these epithelial tumors. HDFSF can promote this phenotypic switch from epithelial cancer cells that are very happy and well attached within their tissue environment to a much more motile mesenchymal-like cancer cell. And this transition is called the epithelial to mesenchymal transition, or EMT, and it's extremely important for cells to escape the primary tumor site and move either through bloodstream or through lymphatic system to different locations where they cause massive problems. And I advise you to read the excellent review, which is with the title Metastasis, Motility and More, that was published in Nature Reviews Molecular Cell Biology in 2003. I've given you the link on this slide. We again see that due to several growth factors and environmental cues, environmental signals, the cells start to lose contact with their environment through to the down-regulation of certain genes and the up-regulations of other genes, and therefore acquire this very motile phenotype, allowing them to move away to different locations where they re-establish a new tumor that attracts new blood vessels and start to proliferate. So there's also the reverse process in which the cells become less motile and start to stay in place and proliferate there. So we're sort of simulating metastasis in vitro in your 96 well plate. We're making epithelial cells change their morphology and start to become motile and move. So what we observe in the assay is a morphological change of the cells. And if you look on the picture on the left, you see the unstimulated MDCK cells that form these very compact and tight colonies where the cells are sort of a cobblestone shape and have intense interactions, are fully in contact with their neighbors. And during one night of stimulation with HDFSF, the cells completely lose contact between each other and start to change shape dramatically and form these sort of fibroblast-like cells, spindle shapes that not only change in shape but also start to wander around in the well. So they start to move away from each other. So what we observe in the assay is a morphological change of the cell, their motility. We could, if we would take time-lapse photographs, you would really see that the cells no longer stay in the same place, but really start to randomly move around. And we see therefore that the preformed tight epithelial cell colonies disperse. So by now you might have the following question, and that is, how are we going to use the assay to understand how we can inhibit the HDFSF MET signaling pathway? And this is a really important question, because we just understood that stimulation by HDFSF leads to colony dispersal and the scattering of the cells, which is what we can very nicely test in this assay. But we want to understand how we can inhibit this. And in our lab, we're very actively working on finding molecules that can inhibit the activity of HDFSF. And those molecules could be antibodies or small molecules or small proteins that interfere either with the binding between ligand and receptor, or prevent certain conformational changes that are really important for the transmission of the signal. In order to test those molecules that we develop, we use the MDCK assay in the following way. First of all, we test the inhibitor on its own on the cells. So you have your preformed colonies, that the 1,000 cells that you plated in each well that formed these very compact small colonies. You expose them to the inhibitor. And this is really important because you want to understand what the inhibitor does at different concentrations. Starting at the highest concentration in, well, A1, let me just get my pointer. And then you dilute them down stepwise all the way to a very low concentration. And you want to see that even at a high concentration, the inhibitor is not killing your cells or affecting the cells in another way. You might sometimes also see that an inhibitor is actually activating cells at a high concentration, which immediately means that you're really dealing with a suboptimal molecule. However, let's put HDFSF in row B. We know what to expect. At a high concentration, we see scattering of the cells, and we slowly dilute again from B1 into B2, from B2 into B3. We dilute HDFSF down to a level where you will see that it stops activating the cells, stops stimulating scattering. And now, with the assay, we take the inhibitor at the different tested concentration in row A, and we mix them with a stimulating concentration of HDFSF. For instance, the concentration you tested in well B3. And I tried to indicate it a little bit with these half wells, where you can see the color of the MET inhibitor being diluted down while we maintain the same concentration of HDFSF in all wells. So it's a mix of inhibitor at a varying concentration with HDFSF at a fixed concentration. And if you have a good inhibitor, you will see that at a high concentration you stop HDFSF from scattering the cells. And you can just go through the wells, because you're diluting the inhibitor, and see at what level, at what concentration the inhibitor loses its potential, its activity to block the HDFSF-MET pathway. And you will slowly see the scattering of the cells coming back. Especially there where you are diluting the inhibitor a lot, you will see full scattering caused by the HDFSF, which is there in its optimal concentration. So this is a very simple example of how we can use to test the MDCK scatter assay to see how well an inhibitor works. I hope this is clear. So normally this experiment, when done by you, would take you roughly 2 hours on the first day, in which you take cells, you trypsinize them. So these are strongly adherent cells that are attached to the flask. You need to trypsinize them. So you use an enzyme to detach the cells. You will count them, and then you plate a specific number of cells in each well that form these colonies overnight. The next day you would come back to prepare your HDFSF dilutions. In other words, you will titrate a specific concentration of HDFSF and dilute it down to a level where it no longer is able to stimulate the scattering of the cells. So that would be done on day 2 and would probably take you something like 2 hours. Then on day 3 we would take the plates out of the incubator, we would remove the media and we would fix the cells with formaldehyde, after which we would stain the cells with a very strong blue dye, which is Comasi blue, allowing us to observe them more easily under the microscope. However, unfortunately, because of the situation we're in, we will do this for you, but we give you this first-person perspective, so it will take a little bit less and hopefully still be very informative and hopefully a little bit entertaining as well. Enjoy! So, are there any questions at this point? Any questions? No? Let's start. So, we have arrived in our tissue culture room. Let's start by switching on the flow cabinet. We switch on the fans and you're lifting the glass to a level indicated on the side. Switch on the light. And then let's do one of the most important things first. We wash our hands. It is important to be as clean as possible, and I will explain later why. We will wear gloves, we'll use some ethanol as well, but clean hands is helping a lot. Okay, and with clean hands we can now also clean the laminar flow cabinet. So we take another piece of paper and spray some 70% ethanol on that. And then we wipe the working area from the back to the front. I mean, this hood for sure is left clean by the previous user, but I think it's a very good way to start your experiment by cleaning your working environment. Okay, so now let's get some important things. You have warmed up your cell medium, the DMAM plus fetal serum. You've warmed up the PBS and you have a tube of trypsin. And I think it's important to place them in a water bath, which is at 37, in order to avoid a cold shock to the cells. So keep everything roughly at the same temperature. Wipe them down with ethanol before taking them into the hood, and then open the cap a little bit just to make it easier for you to handle the bottle or the tube later on when you're holding several things at the same time. Okay, so everything is ready. You're ready to take the cells out of the incubator, so put on some gloves, and please remember these gloves are not sterile and they're really there just to protect you from the experiment. They won't avoid you getting your cells contaminated if you're not working in the right way. You can spray them down with ethanol, but it's just a layer of protection for you. and we're ready to get the cells. So we open the incubator. It's a 37-degree incubator with CO2 at 5%, and inside you find your flask. It's a T75. Let's have a look under the microscope. We switch on the microscope light and let's have a look. Okay, there are the cells. Focus a little bit, like that, perfect. Whenever you sit behind the microscope, immediately put your hand on the focus adjustment knob, and with the other hand you can start moving the flask around, or the dish, or whatever you have, in order to find the cells, because you will see them moving and be able to focus on them. These look like very beautiful MDCK cells. They're nice colonies and the cells look fine. So let's put the flask in the flow hood and switch on the vacuum, which you'll need to aspirate the medium and aspirate the PBS washes. You see there's vacuum there, and there's a nice way of checking it with your finger. So we're now ready to get the cells and start working with them. First thing we need to do is put on some gloves, and then we take the flask out of the incubator. We'll remove the medium and wash the cells twice with some PBS. In order to remove medium that both contains a lot of protein but also contains some specific inhibitors of the trypsin that we're going to use to detach the cells, which are the epithelial cells I talked about in the introduction, that are very strongly attached to the flask. And we need to get these cells into a suspension in order to go into the 96-well plate. So we'll wash the cells with PBS. We add trypsin, which will be a milliliter, cover the surface of the cells and place the flask back into the incubator for roughly 5 minutes in order to detach the cells from the plastic. And once they're detached, we will resuspend them into fresh medium, 10 ml, which also contains fetal calf serum and inhibitors. So the cells are happy in suspension in that tube, but we also stop the trypsin reaction to prevent more damage to the cells. Once they're detached, we no longer need to treat them. We just want to count them. And the counting I'll have a few more words on, because we will use a hemocytometer, and it's always interesting to figure out how we can get numbers from that device. After that, we make a suspension which we pipette into each well of the 96-well plate, aiming to have roughly 1000 cells per well. Okay, so let's start by washing the cells with the PBS. So we first take a Pasteur pipette, a glass pipette, sterile. Everything is sterile which is in the hood, because we don't want to introduce bacteria or yeast or anything. We carefully take off the cap. We've attached the Pasteur to the silicone tube, which is connected to a vessel that is put under vacuum. So we're sucking out the medium that was on top of the cells. Normally I'm trying to reuse the Pasteur, but if it doesn't want to stay there, I just throw it away. Okay, take a 10 ml pipette, like that. Carefully take it out of the package. It's sterile, we have to keep it sterile. We take roughly 10 ml PBS. Doesn't matter how much you take, it's just enough to cover the surface of the flask and the cells, and by moving the PBS over the cells you wash away the medium. I then aspirate, take the volume we put on off again, and using the flask again, we easily get rid of it like that. Throw away the pipette, take a new one, and you know what to do. Normally you avoid spraying the PBS, whatever you introduce into the flasks, directly onto the cells. You sort of hit the back of the flask and then more gently cover the surface by rocking the flask. Well done. So now we take the trypsin. We take a smaller pipette, 2 ml. Again, take it carefully out of the package. It's sterile. We have to keep it sterile. Remove the cap. Take roughly 1 ml, sort of halfway in this 2 ml pipette, and introduce this into the flask. Get rid of the pipette. Close the flask and carefully move the trypsin over the cells. This will immediately start cleaving proteins, among which the ones that are used to anchor the cells to the plastic of the flask. Make sure all areas are covered by the trypsin, and then just place it back into the incubator, where at 37 the trypsin will be most active. Wait 5 minutes. Prepare a little bit the things that you need for when the trypsinization is done. So for instance, take this rack, clean it a little bit. We will also need a tube, because once the cells are trypsinized and detached from the flask, we will resuspend them, and then we will count them. And the counting we do with this device, which is in here, the hemocytometer, which is a glass slide. It's actually sitting in a 20% ethanol solution, so let's just wash it, dry it, make sure it's dry, because it's really needed to allow the cell suspension to be counted properly. And then we need to fish out a little glass cover slip, which is actually thicker than the one you normally use to make microscopic slides. And even though this is thicker, you still have to be careful because it's very easily broken. But it needs to be very straight, and that's why it's so thick. And in order to not lose it, we use these metal clips that keep it in place, just for convenience. Okay, everything clean and dry. Let's put away the box, and it's ready to be used. Okay, and then we need to introduce a small volume of cell suspension in the hemocytometer. So we use the P20, those are the tips. We use a Gilson pipette which we call the P20 because it's 2 to 20 microliters, but there are also other pipettes in there with other volume ranges. And that's it, we're ready.
5 minutes later.
So, okay, let's have a look at the flask. It has been there at 37 for 5 minutes. And let's place it under the microscope and let's have a look. Hey, wait a minute, that looks completely different. Why are those cells moving? And they're all rounded up. In fact, you only see a few that are still attached or partially attached. Let's zoom in, just change the magnification, focus. Yes, indeed, they look completely different, don't they? They're rounded up and many of them are floating. So in order to convince them to really let go, just bash a little bit on the table and place them again under the microscope, and you see more or less all of them have detached. And if you use a lower magnification, you focus a little bit, you will see that more or less all the cells are detached and in suspension. So perfect, ready to stop the trypsin activity by adding a medium. And not only does the medium contain a lot of proteins because of the fetal calcium, but it also contains specific trypsin inhibitors that are present in the serum. So adding that will stop the reaction and will prevent further damage to proteins on the cell surface and keep the cells happy back in the medium. So take 10 ml and add that to the flask. So what you're doing here is just flushing the cells off the bottom of the flask. In case there's still some attached, they will certainly come off once they're well trypsinized. And many of the cells might still be attached to each other. These cells really love to stick to each other and form lumps. So by pipetting it up and down like you're doing now, you really disrupt these clusters of cells, and you slowly start to create a very nice suspension of single cells. And because you used 10 ml, you're ending up with all the cells in that volume. So let's transfer all the cells into a tube. Throw away the pipette. And now it's time to count the cells. So in order to cover this counting area in the hemocytometer, we need roughly 10 microliters. Doesn't matter if it's a little bit more. So we take 10 microliters, make sure that you mix the cells, so the sample you're taking is represents well the whole volume you have there, the whole cell suspension. Introduce it in between the glass cover slip and the hemocytometer, the slide. Capillary force will nicely pull it in, and then you're ready to count. So at this point we have trypsinized the cells and resuspended them in 10 ml in a tube. And we have taken 10 microliters of this cell suspension and pipetted that onto a hemocytometer and exactly onto the slides, the hemocytometer slides, underneath the cover slip, the glass cover slip, which sits on top of the slide. And there's a space there because the glass cover slip is actually resting on the side of the hemocytometer, leaving a little gap between the glass cover slip and the hemocytometer slide. And underneath this area there is a very, very fine, almost microscopically small grid, which consists of scratches, actually, very nicely drawn lines that give you a very nice black line with a very light, transparent background of the glass hemocytometer. And this grid is very accurately made and allows you to measure the number of cells in a certain area. The 10 microliters of cell suspension flows over this grid. It's sort of pulled in by capillary forces and deposits the cells, if everything goes well, in a sort of random and well dispersed way on top of this grid. And because we know exactly the surface area of the grid, which for the corners is 1 by 1 millimeter, and we know the height underneath the glass cover slip, we can understand what volume is actually in each of these corners. So what I normally do is I count the number of cells in the corners. There are also people that count cells in these other areas which have much finer grids, but I just count the corners. And I always count 2, and if I find that the number of cells that I count are quite equal, I count 2 and no more. However, if I find some deviation, I count up to 4 in order to get to a proper average. And now we go back to our surface area, which is 1 by 1 millimeter, 1 by 1, with the height, which is 0.1 millimeter, which actually gives us a volume of 10 to the minus 4 cubic centimeter, which is equivalent to 10 to the minus 4 ml. Therefore, in order for us to understand how many cells we have per ml, we multiply by 10,000 to make up for this 10 to the minus 4. So if we have 5 cells there, 7 cells there, we take an average which is 6, we multiply this by 10,000, so we have 60,000 cells per ml. That is, in the way, the number of cells per ml, sort of the concentration of cells, if you would like. If you want to know how many cells you had in your flask, remember you trypsinized all the cells and you resuspended them in 10 ml. So the total number of cells here, you counted 60,000 cells per ml. You have 10 ml in total, so you have 600,000 cells in total that were in your flask and are now sitting in your tube in a volume of 10 ml. This will allow you to do further calculations in order to understand how you can take 1,000 cells into each of the 96 wells. I hope this is clear. So let's start placing the hemocytometer under the microscope. Again, get the calculator and the counter. Make sure it's set to 0, like that. And now it's just simply a matter of counting. I wanted you to have a look at the grid, but we know what we are looking at. And there you go. Yes, and sometimes with this you really wish you have fewer cells per ml because it's just quicker. However, of course, the important thing is that you have enough cells in suspension to do your experiment, so you need to get it right. How many did you count? 117, okay. So let's write that down on the glass screen. And then let's count the other sides as well. So reset your counter and move to a different corner of the grid. Alright, you're already finished. You counted 4 corners, and what you counted was 117, 148, 103 and 133. So just take the average of that. How much is that? Okay, 125. So how many cells do we have per ml? Indeed, times 10,000. So we have 125 times 10,000 cells per ml, which is 1.25 million cells per ml. And that sits in the 10 milliliters of cell suspension in your tube. Okay, now the important part. How much do we need to prepare? How much of the cell suspension do we need? So if we say that we want to introduce 1,000 cells per well, and we want to introduce that in 200 microliters, if 200 microliters contains 1,000 cells, then 1 ml would contain 5,000 cells. We will roughly make enough for 100 wells just to have a little bit of extra volume. So let's take 5,000 divided by 1.25 million times the volume that we want to make, which is 20 ml, gives us 0.08 ml, which is 80 microliters. So we need to introduce 80 microliters of our cell suspension into 20 mLs in order to have a concentration of 5,000 cells per ml. Okay, so let's get the 96 well plate, which is sterile, get it out of its package. There's a left and a right side, and you recognize the left side because it has the corner. On the left side you also see A, B, C, D, E, F, G, H, and on top 1 to 12. Label it. Make sure you have your name and date on it. Maybe even a description of the experiment. And then, how much did you calculate you needed to introduce? Correct, 80 microliters. So the P20 that you have is no good. We need to get the P200, which goes from 20 microliters to 200 microliters. Get everything ready. You have your cell suspension. Adjust the pipette to 80 microliters. Take a tip. Everything is sterile. Be careful with it. Mix the cell suspension because the cells will slowly sink to the bottom. Take 80 microliters. Add that to a large 50 ml tube. Pipette against the wall, so you make sure you have the whole 80 microliters. And now let's add 20 ml. We take a 25 milliliters tripette. Take the medium. And add the 20 ml of medium to the tube. Mix a little bit by pipetting up and down. And that should give you a cell suspension, 20 ml cell suspension of 5,000 cells per ml. Okay, now let's have a look at this funny thing. It's also a pipette, but it's a repeating pipette, and it uses this tip, which is called a combi tip, and the volume of that is 5 ml, and it also says 1 means 100 microliters, setting 1. So it's sterile, so carefully peel it out of its package, insert it into the repeating pipette, and then set the dial to 2, because 2 times 100 microliters should give you a pipetting volume of 200 microliters, and the display will indicate that as well. Mix your cells, and then pull up the plunger like that to fill it, and then press the lever, the dispensing lever, once to reset it. Then you're ready to dispense 200 microliters with each press of the lever, like that. Okay, and you should be able to do 2 rows with this. But when you get to the end, be careful, because the last one might not be the full 200 microliters. Empty it, fill it by pulling up the plunger, reset it so the volume is accurately set, and there you go, dispensing 200 microliters each time in each well.
Boring!
Yes, indeed, this can get a little bit boring, so sometimes when no one is around, you start to race with yourself, see how fast you can fill those wells, and this often ends with cells splattering all over the place. However, we are reaching the end of the plate, which means we're almost ready to go home. Funny thing is that even though we made 20 ml, which should be enough for 100 wells filled with 200 microliters, you always end up with a little bit less. There's some foam, there's some loss here and there. Anyway, we don't need all the wells, we're only going to do the top row. So this is it. Put the plate back into the incubator and see you tomorrow.
The next day.
Yo, welcome back to the lab. How did it go yesterday? Was everything fine with the cells and the counting? How did you manage? I think today we're going to find out, because let's take the plate out of the incubator, place it under the microscope and have a look at the wells. What happened during the night? Did the cells grow? Did they form colonies? We don't need to do all 96 wells, that's rather boring, but you just need to get an impression of what happened there. Theoretically, they're all the same. Our biggest fear is that during the experimentation yesterday, during the preparation of the cell suspension, some uninvited guests arrived. Yeah, today I'm going for the big red swimming pool. Exactly. Those nasty bacteria or yeast, they're everywhere. And the moment we start working, even in a flow hood, which provides you a rather clean and sterile environment, it's still difficult sometimes to keep them away from your experiment. So check those wells, see if the medium is still clear and the cells are happy, and you'll probably see colonies consisting of 3, 4, 5 cells, rather compact, and that's really what we want. And of course those cells are okay. I had no doubt you did an excellent job yesterday, and it's a right moment to move on to the HDF-SF dilutions. However, I do think it's wise to check the plate just to understand whether everything is fine before making these dilutions, because it takes time and it consumes protein, medium, and takes consumables such as the Eppendorf tubes. So in case you find yourselves to be contaminated, unfortunately, or you find that somehow the cells did not attach well or form colonies, this is the moment to stop your experiment and maybe set it up again. However, everything looks fine, so let's move on to the HDF-SF dilutions. The first thing we do is we take from the -80 a stock solution, and we find that written on the label it says 0.7 milligrams per ml. And it's the concentration given in milligrams per ml is quite standard for proteins in solution. However, in our experiment we talk about molarity, and if you remember correctly, it's rather simple to go from mix per ml or for grams per liter to a molarity. You simply need to divide by the molar mass. If you remember the Avogadro number 6.0221407.6 etc. times 10 to the 23rd, a rather large number of molecules give you this weight in grams per mole. And for a large protein like HDFSF, this is a rather impressive number. It's 79 kilos per mole. Can you imagine? However, to understand how much we have in this tube, which contains 0.7 milligrams per ml, we simply need to divide. We go to, first we change to the volume that we normally use for molarity, which is a liter. So 0.7 milligrams per ml is 0.7 grams per liter. How many moles are in there? Let's divide. 0.7 grams divided by the molar mass, which is 79,000, gives us a molar concentration of 8.9 times 10 to the minus 6, and that is 8.9 micromolar. So we move to what we need for our experiment, which is in the first well we wanted to have 10 to the minus 8 molar. So we understand we have to lower the concentration even further, so we need to dilute. We need to take a small amount of the stock into a larger amount of medium in order to bring the concentration down even further. And for convenience, let's make 1 ml at 10 to the minus 8 molar. So in order to understand how little we have to add to the 10 ml to dilute it down, we just divide 10 to the minus 8 molar by the concentration that we have in our stock, which is 8.9 times 10 to the minus 6 molar, and we multiply that by the volume that we're going to make, and we get to this tiny little volume, which is 1.12 microliters. So the only thing we have to do is add 1.12 microliter into 1 ml and mix it very well, either by rocking the tube by hand or by vortexing. And when we come to dilutions, we really figure out that it's the ultimate way to make people fall asleep, not impress your family and friends. So we need to quickly come up with something that makes it sound much more complicated than it really is. What about semi-log scales and half-log dilutions? That will impress anyone that is not really trained as you are. So let me show what we're going to do and how you can impress your friends that are doing other things than following the Harvey course and becoming doctors. You see this chart, which is a semi-log scale chart. You probably had it before in different courses, but on the x-axis you have a logarithmic scale taking the concentration down in steps of 10 times dilutions, and on the y-axis you have a range of activity. This is all imaginary. It's an imaginary experiment from 0 to 40. So very easy. If we want to make dilutions in order to test a particular protein, for instance, and see what its activity is on cells, we can just go for straightforward 10 times dilutions, and that's an extremely efficient way of bridging or reaching a very high range of concentrations in a rather limited number of steps. We go from 10 to the minus 8 molar to 10 to the minus 9, 10 to the minus 10. We can go all the way down to 10 to the minus 12. We reach the picomolar range in order to see where our protein stops having its effect. But what if you want to do a step in between? So a step in between, let me get my laser pointer, in between this 10 times dilution. We're so used to a normal scale from 1 to 10, 10 to 100, where if we go halfway, we just divide by 2. So we end up with 5 times 10 to the minus 9. But if we plot 5 times 10 to the minus 9, you will end up in this spot here. And on a logarithmic scale, you're actually off-centered. You're not in the middle. So in order to get halfway in between these two concentrations, wherever you are, we actually need to do a half log dilution. So we have 10 to the power of 0.5, which is indicated here, which is a 3.162278, etc. fold dilution. And if you do that, so if we ignore this 5 here, we go from 1, we go to 3.16 times the lower concentration. You place the value beautifully in the middle, in between the two concentrations. And that's what we're going to do. So I hope this is clear. It's just to accommodate the logarithmic scale and the logarithmic dilutions, where we go halfway, and that is a 3.16 times or fold dilution. So how we're going to do that, we go again back to our HDFSF stock at 0.7 mg per ml, and we just calculated in the slide before that we have to take 1.12 microliters, a tiny volume, into our first tube, which contains 1000 microliters, in other words 1 ml of medium. We mix well, and then we take 100 microliters, which we add to a volume of 260 microliters of just medium. We mix again, so we have a mixing ratio of 100 divided by 316, which is this 3.16 fold dilution. We mix, we take again 100 microliter, add it to 216 of just medium, and we go on as long as we want to get to, well, down to the picomolar range, as far as you want to go, really. And then we have all these tubes. They all contain roughly 260, and just a little bit more than 200 microliters, of course, except for the first one, which still has 900 microliters in it. We take 200 microliters of each tube into each well, so we have the whole range of concentrations on the top, and that's it. That's it. We just remove the medium in which the cells grew overnight and replace it with our dilutions, our diluted HDFSF. So enough talking, go and prepare those dilutions. Replace the medium on the cells with your new dilutions, which is always fun to do with the vacuum, because the sound it makes really reminds me of going to the dentist, which actually is maybe not that funny, but okay. Place the plate back in the incubator, and guess what? You are finished for today. Oh, let's first start switching on the flow cabinet. You remember we lift the glass screen all the way up to the level indicated on the side, so the beeping will stop. The fans will rev up, but you have to pay attention to these red lights because it means that the flow has not yet been established. Switch on the light, and then we do our normal boring routine. We wash our hands. Let's clean the flow cabinet. Take our medium out of the water bath. This time we don't need the trypsin, we don't need PBS, we just need plain medium. And then we need, of course, a lot of tubes. So a tube rack is very handy. The Eppendorf tubes are in this yellow box. And we need a pipette, so we need tips, the P1000 and the P20 or P200 tips. So we get the appropriate pipette, Gilson pipettes from the drawer. Organize it a little bit. A pen. Okay, and then let's quickly make ourselves a little overview of what we're going to put where. So we have a nice template for the 96 well plate, and today we're just going to pipette an HCFSF dilution in there, but you can imagine you could use many more rows to test inhibitors or stimulating ligands. It's really up to you. And then we do the calculations as we discussed also in the with a few slides that I introduced. So we have HDFSF and the stock concentration is 0.7 milligrams per ml. I'm sorry that that's just out of view. The molarity is therefore calculated by looking at how many grams you have per liter and dividing that by the molecular weight. So we have 0.7 milligrams per ml, which is 0.7 grams per liter. 0.7 grams divided by 79,000 grams, which 1 mole of HCFSF would weigh. Okay. So just let's get the calculator. You already know probably where we're going with this. Correct. 8.9 times 10 to the minus 6 molar. So the concentration in the tube is 8.9 micromolar. So we want to start with the first well at 10 to the minus 8. So let's see how we're going to dilute. Again, this is repeating what we already discussed with the slides. We have 10 to the minus 8 molar, what we need. And so we divide by the current concentration, which is 8.9 times 10 to the minus 6, which is actually a much higher concentration. And here you write 350 microliters, but in fact that would give you a pipettable volume of, let's see, oh come on, very, very, very small volume, 0.39 microliters. I'm actually not so comfortable pipetting that accurately, so it's much nicer to pipette a slightly bigger volume. In fact, with these things, if you want to be very accurate, you want an even bigger volume than we're going to use, which is a volume that we pipette into 1 ml, 1000 microliters, which we figured out was the 1.12 microliters. Okay, let's take the tubes that we will need for the dilution series. The tubes come from this box, which is autoclaved, so the tubes which we call Eppendorf tubes, which can contain up to 1.7 ml, normally we call them 1.5 ml Eppendorf tubes, are sterile. So in order to keep them sterile, which is essential for the experiment, we handle them very carefully. We do not touch the caps, we do not touch the inside of the tube, because even though you're wearing gloves, those gloves are certainly not clean anymore, because you're touching all sorts of surfaces that are not sterile, and it's very easy to introduce bacteria if you're not careful. So we know we need 1.12 microliters of HDFSF stock in 1 ml of medium. And in order to get the 1 ml from the flask, you could use the P1000, very accurate. However, that pipette is rather dirty because only the tip we put on is sterile, coming from the blue box. And you would have to go in with the pipette past just the tip. So it's much easier just to get a volume a little bit over 1 ml with a very clean and sterile stripette, like you do now. So place that there, get rid of the stripette, and then use the P1000, which is for sure more accurate to get your 1000 microliters into the first tube, in which you will dilute your HDFSF down to 10 to the minus 8 molar. And then we're going to introduce the 1.12 microliters, the tiny, tiny little volume. Here you have your stock of HDFSF. So 1.12 microliters. It's almost not visible in the tip. Goes into the 1 ml. Sometimes you check a little bit if everything came out. It did. And then it's a matter of just mixing very well. And the vortex does a great job at that. Inside the tube you can see like a little mini tornado if you're doing it well. It's very rigorous mixing. Okay, let's have a look at what we need. We have the new HDFSF stock, which was a dilution of the stock we took from the minus 80. So it's at 10 to the minus 8 molar. And you have 1 ml of that. So, okay, the dilutions, we know what to do. We take 100 microliters from the first 10 to the minus 8 tube into the next tube, which contains 260 microliters of DMAM. And with that we have the half log, the 3.16 times dilution. Let's take some DMAM, and again we're not using our pipettes to go inside the bottle. We use a clean stripette, which is sterile in its package. Carefully take it out, and we take a rather large volume, maybe even more than 5 ml, in this case 7 ml, into the tube, and we pipette from there. So we don't have to open and close the bottle with medium all the time, and we don't risk contaminating it. So 216, we need the P1000 for that. We cannot do it with a P200 because it's just over the volume that it allows you to pipette. So 216, I don't know if you can see that. We take a blue tip and we start filling the tubes from A2, A3, all the way down to A12. So, the moment we've done that, we go back to our P200 and we set it to 100 microliters, because now we're going to do the actual dilutions. The 100 microliters that go from the left tube into the right one. So 100 microliters into 260 microliters of medium. There you go, 100 into 216. So you have 316 in total. Vortex, mix really well. Take a new tip and then take from that mix into the next tube. 100 microliters into 216. And vortex.
Many minutes later.
Once you're finished with all the dilutions, let's also take a volume of medium that we will put on the control cells, and those will be in row B. You do want to replace the medium there, even though you're replacing it with fresh medium that doesn't contain anything extra. There's no HCFSF in here. It is the best control that you can have with your stimulated cells, in which you also replaced the medium in which they grew overnight. So it's really important to have this perfect control. Okay, we're going to replace the medium that's on the cell, so we have to switch on the vacuum and of course check as well, just for fun. That works. Okay, go and get the cells. You can indeed put a little bit more ethanol on your gloves, just have it extra clean. And let's go to the incubator to retrieve the plate. Does everything look okay? I know you already checked, but still, it never harms to check again. Cells look fine. Okay, we're ready for the next step. Okay, organize a little bit your working space. Everyone has its own sort of layout or setup. Make sure you have your space so you don't have to go over the plate with your sleeves or hands or tubes or anything. Just organize it a little bit. We have our row of dilutions. Let's take a clean Pasteur pipette. This is glass connected to the vacuum, and that will allow you to aspirate to remove the medium. So let's go for the top row. And this is what I said, reminds me of the dentist. Okay, the cells are now exposed to air. They really don't like this very much. So it's important to quickly get the medium on top of the cell. So set your pipette to 200 microliters, take a new tip and start pipetting those dilutions onto the plate, into each well. 200 microliters at 10 to the minus 8 molar in A1, new tip, A2, 3.16 times 10 to the minus 9 molar, and so forth.
Boring. You're boring everybody. Quit boring everyone.
That looks really beautiful. You've also done the second row, row B, with the control medium, just medium with fetal calcium without addition of anything. And now the cells are ready to scatter overnight. That was it for today. Last thing to do is just to clean up and leave the hood, the laminar flow hood, in a very clean and nice way for the next user. So we just wipe it with ethanol. Maybe we should also remove the writing. Switch off the vacuum and get rid of those gloves.
Many unbearable hours later.
This will be it. This is it. And when I say this is it, it really means this is it.
Hey, hello, welcome back to the lab. This is it, day 3. This is the day in which you're going to see the result of your hard work of the last 2 days. All the tedious pipetting, the dilutions you did yesterday, and the cell seeding the day before. Today we're going to see the results of that effort. And therefore, let's not wait any longer. Get your plate from the incubator and place it under the microscope. And what do you see? Possibly you will see this image, especially when you're on the left side of your row A. You will probably see these very nicely scattered MDCK cells. This also means that you had no uninvited guests in your experiment. There are no bacteria, no yeast, which sometimes are actually very difficult to see because they are tiny. Especially bacteria are not easily seen under this stereo microscope. But you will see the effects on the cells. You might see that the cells look rather stressed or the medium has changed color because the bacteria have acidified the medium. Both the secretion of toxins and the change of pH really affects the cells, and you might get some very strange results in an assay from a contamination. So it's something you have to pay attention to. However, the cells I think look really nice. So the next thing we want to do is we want to fix the condition as it is now. So you want to have the cells more visible, and that's something we can do with a Kumasi blue dye, but we don't want to change the morphology or lose the cells by treating them. So what the first thing we do is we get rid of the medium. We throw that into a box and decontaminate that medium in case there are any loose cells still alive. We fix the cells with this 4% formaldehyde solution in each well, and then we get rid of this formaldehyde solution and submerge the plate into the dark blue dye, the Kumasi blue stain, and leave it there for at least an hour. Also the fixing we do roughly for 1 hour. And after that we wash under the tap the plate. We do that relatively careful, even though everything is fixed and the cells are pretty well attached to the plastic. I still don't want to rinse them off, so wash the plate, but be gentle doing that. And then slam the plate on a piece of paper to get rid of excess water. And if you want to dry the plate very quickly, just place it in an incubator, 37 or even warmer. After which the dry plate can be placed under the microscope. You no longer have to keep the lid on because of course everything is fixed and dead. And then you just observe the cells, and they will look like you see here. They're beautifully blue. And then what's left to do, which is really important, is to score the plate. And what I normally do, I have a good look at the control cells, which you placed in row B, which will look something like this. Very tight colonies where you can almost not see the borders between different cells. That's a clear negative. And then you look at your HDFSF treated cells, and you might go from left to right, but some people also go from right to left, scoring whether you see a positive scattering of the cells or a morphological change. And then it's really up to you to determine at which concentration you no longer see this effect. Where there's the turning point or the cutoff point, at what concentration. And I normally score the scattered cells as positive, the non-scattered cells as negative, and somewhere at some concentration I find maybe a sort of dubious scene of cells that are close together but still are morphologically different from the control cells, and those I call plus minus. Sometimes you have a really sharp cutoff, which means that you go from one well being at a positive scatterer and the next is really negative. It can happen. The most important to realize here that this is a sort of qualitative scoring that you do, and people have different opinions about this. In other words, if you give the plate to someone else, he or she will have a look at it and might place the cutoff point, a plus minus, in a different well and at a different concentration. However, when you are doing your experiment, you will include in your plate the right controls, the positive controls, the negative controls, and you will do a comparison within the plate, within your own experiment, and you as an observer will therefore be able to make some proper statements about the activity, for instance, of an agonistic molecule, or the inhibiting activity of an inhibitor that you developed that blocks the activity of, for instance, a challenging HDFSF concentration. That's it. Take your notebook or take the template that you made where you have the different concentrations written down and start scoring. Look under the microscope and give the wells a plus minus, or a minus, and in some cases I see sort of different scattering. Sometimes I see super scattering, which I then give a plus plus. It's really up to you, but I'm very much looking forward to see the results. Very curious. Enjoy.
2 hours later.
Yeah, I'm sorry, I have to apologize. I was not there when you fixed the cells for 1 hour and you then placed the plate into the Kumasi blue for 1 hour. I had to be somewhere else, I'm sorry. However, I see everything went well, and okay, you're just getting rid of the Kumasi blue. Indeed, try to get as much in the box out of the plate. And then use some water, some distilled water in this case, to just rinse away the remaining blue. I'm always trying to block a little bit the flow of water from the tap to not go directly onto the cells, but they are pretty fixed and adhered, so don't worry too much. And then you will see that in fact the plate remains quite blue, and actually that's because of the stained cells. That's a good sign. Get a piece of paper, let's wipe away all spots of Kumasi, dry the plate, just slam it, you get most of the moist out of the plate, and that looks really beautiful. Let's remove all evidence that we have ever been in this room, and let's take the plate to the incubator. So at the end of our corridor we have an incubator room, and there's this stove, this incubator, which is at 37 degrees. Just take the lid off and leave the plate in there for as long as necessary.
A few inches later.
Yes, it really doesn't take that long to dry the plate often. So now is really the moment we're going to have a look at the result. I'm extremely curious. Let's go back to the tissue culture room. Okay, let's take the plate, take off the lid and place it on the microscope. And there we go. Make yourself comfortable and start looking at those wells. Maybe first with a low magnification, followed by a higher magnification. What do you see? Okay, well A1, beautifully scattered cells. Focus a little bit, keep moving the focus knob, the adjustment. And let's have a look at the next well, A2. Again, beautifully scattered cells. You see many of them ended up away from the center, which is often happening. But all of them look very nice and scattered. A3, also beautiful, very little background staining and clearly activated by HDFSF scattered cells. A4, a very similar story, nice scattered cells. Okay, now let's zoom in a little bit on the colony. So change the magnification, increase the magnification. Keep adjusting the focus plane and move through the well to see all the cells, to see all the colonies and get a general overview of the state of activation, cell activation. They look very beautiful. Okay, record your observation. A1 was definitely positive, so put a plus in that well. Go back and keep checking each well. A6, again positive scattered cells. Some are very funny, they're much bigger, little pancakes, but the overall idea is really that they're well scattered. Keep changing the location with the platform, XY platform, and keep observing them under high magnification. And here in A7 you actually see that the cells are slowly getting closer together, but there are definitely different regions. Some colonies are very compact, others are still very scattered. So you have to start getting an impression of what's happening. Clearly still scattered. Make your markings on paper. Record your observation. Go back to the wells. Okay, we ended up in well A8, and there really I think the colonies are much more compact, but make sure that you observe most of them. Move around through the well because you see different regions and different cells behaving in different ways, but clearly very compact phenotype. So I would call that maybe a negative. Here we end up at well A9, not so different from A8, so difficult sometimes. So the best thing to do is to move to the control well. So let's have a look at B9. Yes, that looks very, very similar. So for sure A9 is a negative well. So something is happening around A8. So let's conclude your observations. You've looked at all the wells in row A and probably at a number of wells in the control row B, where the cells are not scattered, have not seen HDFSF. And you probably came to the conclusion that on the left side in row A, well, A1, A2, A3, A4, A5, A6 for sure, and A7 are probably showing scattering. So let me get the pointer. So you mark those wells as positive, and then somewhere near well A8 and A9 something is happening. So let's have a look at well A7. Okay, at well A7 we start to see that the cells come closer together and start to form colonies, but for sure there's still a lot of space between the cells. They're not as closely attached to each other as you would see in row B. So let's compare with well A8, and there we see clearly a sort of different phenotype. We see very compact colonies with very little space in between the cells. In some cases it's almost impossible to distinguish single cells within a colony. And how does that compare to well A9? Okay, I think well A8 and A9 look pretty similar to me. We see very compact colonies in A9, and clearly there's no observable scattering there. It's important to look throughout the well, so to really see whether your opinion on that concentration of HDFSF and its effect on the cells is really not biased by a number of very remarkable colonies that you look. You really have to make sure that you look at all the cells in a well, and we've seen under the microscope that you can find sometimes very differently behaving cells and colonies. So how does well A9 compare to well B9? Well, you really see that B9 and non-stimulated cells form these very tight colonies, but in my view they are pretty similar to A9. So as you did, A9 is negative, as are the cells in row B. So you can conclude that well A8 is probably where you lose activity, and you can label it as plus minus. Sometimes I would write plus dash minus minus to show that we're really very close to losing all the activity. It really doesn't matter much, but we have to understand that maybe someone will say, no, this to me already looks like negative. It's a non-quantitative assay. It's really a qualitative assay where you as an observer establish where this cutoff point is, and really use internal controls or internal comparisons within the assay to make statements about the potency of either an agonistic molecule that you could use, for instance, in regenerative medicine to heal organs and to heal epithelial tissues after damage, or as we were saying in the light of oncology, where you test an inhibitor and see how you can block the activity of HDFSF at different concentrations. So as long as you stay within the same assay and you as an observer judge all the activities that you can see, the scattering activity caused by HDFSF or an agonist under the influence of an inhibitor, you are able to make comparisons. And for this, this type of non-quantitative experiment is extremely important. Okay. I think this brings it to an end. You have beautifully marked and recorded your observations and established the cutoff point at 3.16 picomolar HDFSF, which is really what we often get in the lab. Very beautiful assay, extremely sensitive. That was it. Thank you very much, and let's move to the last slide. So with this, we've come to the end of this tutorial, and I really hope that you enjoyed doing the experiment quasi yourself in this first-person format. We are very sad that we were unable to offer you a true in-person experience, as we believe that tutorials should be an essential part of studying and your training as a doctor. We are convinced that this experience can enrich the subjects we teach in class, and we actually noticed that the moment you start performing the experiment yourself, questions arise that you would not have while studying from books or articles. And most importantly, a tutorial allows you to understand the intricate connection between experimenter, experiment and outcome. And there's no better way to explain what science is truly about than to perform experiments yourself and understand how results are obtained and knowledge is acquired. I therefore hope that as soon as this crisis is over, you will have or will search for other opportunities to do experiments. And then finally, I would like to thank Dexter for his contributions to the tutorial, as well as the opportunistic yeast and bacteria for not interfering with your assay. And that's it really. For now, I wish you success in the coming months with your coursework and especially with the examinations at a distance. Take care and stay safe.